So thank you very much for inviting me to come here and talk to you about what we've been doing to relate variation in the host genome to variation in the microbiome in two systems. So I'll be talking about humans, where we look at the gut microbiome, and in Maize, where we look at the root-associated microbiota. So I'm going to start with the human microbiome. As you know, there's 10 to 100 trillion cells that live on and inside our bodies, and they encode 150 times as many genes as the human genome. The vast majority of these live in the large intestine, and here they help break down fiber. They produce vitamins, they degrade xenobiotics, and so on. And the gut microbial communities, which are shown here in this pie chart, have a particularly distinct composition compared to those of other body parts. They tend to be dominated by firmicutes and Bacteroidetes and some actinobacteria, and comprise somewhere between maybe 500 or 1,000 distinct species, types of bacteria. And there's a growing body of work showing that an alteration in the community composition can be associated with different disease states. So for an increasing number of diseases, there's what we call a dysbiosis, or an alteration of the lineage content of the microbiota. Here are some examples. For instance, in inflammatory bowel disease, patients tend to have a proliferation of proteobacteria in their guts. Type 2 diabetes has also been associated with a shift in the dominant phyla. Here's some data for necrotizing enterocolitis in infants, where again you have a proliferation of proteobacteria in the disease state. And these are just some examples. There's an increasing number where the dysbiosis is associated with disease, and that begs the question of whether this is a manifestation of the disease or secondary, and if these alterations in community composition have something to do with either predisposing the host to disease or contribute directly to the disease state. So we and others, and my work with Jeffrey Gordon, included this component, which was to take germ-free animals as a tool where you can directly test the impact of a microbiota by introducing a microbiota from, say, a healthy host and one from a diseased host into animals that are born and raised under completely sterile conditions, so they lack a microbiota in these sterile isolators. And so now for a number of different diseases, these experiments have shown a direct causal effect for the microbiota of the gut on different diseases. So for instance, we worked on non-obese diabetic mice that could be protected against the onset of diabetes by a particular microbiota. Work with Jeff Gordon that we did on obesity showed that obese-associated microbiotas led mice to gain excessive weight compared to microbiotas from lean animals. More recently, and Rob mentioned this yesterday, we've worked with Andrew Gewurtz at Emory University to show that a particular microbiota was sufficient to cause metabolic inflammation in hosts, even genetically normal hosts receiving a particular microbiota develop metabolic syndrome as a result of metabolic inflammation and insulin resistance. And then Laurie Glimsher and Wendy Garrett at Harvard have shown that a particular microbiota can cause colitis. So together, these studies are starting to really show that the gut microbiota can be really important, not only as a biomarker for health, but also can be directly involved in some of these chronic inflammatory diseases. So we're particularly interested in how microbial communities form and what determines their composition. We've looked at the developing infant gut, and this is an example of one individual who was followed over 2.5 years. So as you know, babies are born germ-free, and Rob showed nice data yesterday showing that the very initial colonization comes from exposures to the microbiota of the mother and the people around the mother. But then that initial colonization process changes, and you see, so you're looking here at the distribution of different phyla at different time points from birth to 2.5 years. And there's certain events, like here, for instance, was a fever event that led to a transient change in the composition, and these bars here that change to orange indicate Bacteroidetes really coming into the system after solid food came into the system. Not so much rice cereal. It turns out that the microbiota are ready to handle rice cereal, and that really has no impact whatsoever. But you start putting other more complex glycans into this system, and you see a big shift in the dominant phyla, and then that brings about a shift in the functional genes as well. So here's some metagenomic data for some of these time points, and the shift in Bacteroidetes is accompanied by an enrichment in some of these glycan-degrading genes, as you can imagine. So even though you see these nice big shifts in phyla composition that you can attribute to events in the life of a developing infant, when you look at the species composition, and now we're looking at the same time series, but each one of these dark areas shows abundance of these particular taxa that are shown on this tree here. So you're looking at the abundance of different taxa across the samples, and you have an early set of species here, and then there's a slow succession over time where these different assemblages succeed each other. Here, for instance, there was an antibiotic treatment where some of these lineages that were apparent early on came back, and then a shift right after that to a whole set up here that hadn't really previously been seen. And so we can see that certain events engendered these shifts in composition. Even though many of these are still Bacteroidetes and Firmicutes, they're clearly a different set of Bacteroidetes and Firmicutes. So there's the subtleties at the species level here in these consortia. And we can tell by looking at co-occurrence patterns and co-exclusion patterns that these are really non-random associations. Compared to random distributions, we see that some types always co-occur and sometimes some types never co-occur. And this fits very nicely with what Peter Brook showed yesterday, that there are certain enterotypes, and I think that's what we're seeing here. We see certain consortia that clearly work well together, and you can shift a system sometimes from being dominated by one to maybe dominated by another. But at this point, that's what drives these changes in microbial diversity, and these shifts to one group to another is still not very well understood. But the pattern that's emerging is that there's a core microbiome, or core lineages that are shared amongst all of us. That's a relatively small fraction of the microbiota, of the lineages, and then we share functional genes as well. But then there's this variable microbiome of lineages and genes that varies between people and varies between health and disease states, and that can be influenced by a number of things, from the host environment to the host diet to the host lifestyle to host health, and also potentially to host genotype. And the reason these arrows are all the same shape or the same size is that we really don't have a good idea of how which ones are more important or how they interact or any of that. But what we're looking into is this one here, how the host genotype can affect the composition of this variable microbiome between people. The work that has looked at host genotype and host genetic variation on the composition of the microbiota has focused largely on candidate gene approaches. For instance, there's a study in humans that looked at familial Mediterranean fever, which is caused by a single SNP in a gene, and individuals carrying that SNP have a decreased microbial diversity in the gut. And then there's a number of animal studies where a single gene is either knocked in or knocked out, and people have looked for effects. So again, some of the work we were involved in looked at loss of toll-like receptor 5, which has a big impact on the microbial diversity, and that's linked to disease. Or in type 1 diabetes, in the non-obese diabetic mouse model, loss of MyD88 results in a change in the microbiota as well. And there's a number of these model systems where people have looked at the effects of particular genes. And there's one very recent paper by Daniel Pomp and colleagues, which actually took a quantitative genetics approach in mice and used an advanced inbred line of newly derived mice to look for genetic effects across the genome and look at effects on microbial community in general. But there haven't been, so far, any genome-wide analysis studies in humans linking human genetic variation to variation in the microbiota. What there has been is some studies with twins, and this Rob showed yesterday. This is one of the twin studies that I was involved in with the Gordon lab, where a small number, a relatively small number of twin pairs, so 31 monozygotic or identical twins and 23 dizygotic or fraternal twin pairs was studied, and one of the goals was to see if the monozygotic twin pairs had more similar microbiotas than the dizygotic twin pairs, which would tell you that there's a host genetic effect there. And there was a trend, but it wasn't significantly, there wasn't a significant difference in these unifrac distances. So if there was a host genetic effect, you'd expect the MZ bar here to be lower than the DZ bar, but that wasn't apparent with this data set. But what we're doing is using twins again, but genotype twins. So we're collaborating with Tim Spector in King's College London, who runs a twin registry, and Andy Clark at Cornell University. The twin registry has 8,000 twins, and they've all been genotyped. They're half MZ, half DZ, and they have full health records for these individuals and then a number of other data sets for these individuals. So what we're doing is taking the genotype data and the health records, and we're sampling feces from these people and generating 16S data, plus some other measurements in stool, starting with a pilot study of roughly 600 individuals. And to just run you briefly through the pipeline of how we analyze these 16S genes, we've been doing this with titanium. We use Rob Knight's software, CHIME. We generate 16S amplicons. We can keep track of what sample each one came from by barcoding them. Once we have the data, the sequence data, we can bin sequences into operational taxonomic units and keep track of representative sequences. We can then have OTU-based approaches where we keep track of how many sequences of each OTU were found in each sample. We can build phylogenies and then do phylogeny-based analyses such as Unifrac, which Rob talked about yesterday, to analyze these data sets. So this is very preliminary data from our first set of twins, 24 monozygotic twins and 19 dizygotic twin pairs. And this is the same analysis again that the Turnbow 2009 paper performed, computing Unifrac distances, which give you an idea of how similar communities are overall using the phylogeny information. And here we see in this group a significant difference between MZ and DZs, where the MZ twins have more similar microbiotas than the DZ twins. And so this is telling us that there's something here under host genetic control, there's heritability, and there's something to go after to find out what host genes could be driving this. We used the SNP data, especially from the DZ twins, to infer identity by descent across the genome. So when you have two siblings, they share half of their genetic information on average, but that's actually variable. If you scan across the genome, some regions you can have more of the genome that's shared between two siblings, and some you can have less. And this is data from Peter Vischer from his twin cohort showing the frequency of how much of the genome is shared, and for the most part, half of it's shared between sibs, but then there's some parts that are more shared and some parts that are less shared. So what we could do with our twins is go through and look at sliding across the genome how many alleles are shared, either 0, 1, or 2. And we get patterns like this. So now you're looking at IBD sharing across a genome. These are the chromosomes here. They alternate by color. And you're looking at whether twins share have no alleles in common, or 1 or 2. And as you slide across the genome, you can see whole blocks where they either share 0, 1, or 2. And these are different twin comparisons here. So what we're doing is these regressions where we take the IBD sharing And we use those in regressions against the quantitative data that we get from the microbiota. So for instance, abundances of particular microbiota. And so we're regressing the IBD sharing against the differences for twin pairs in these quantitative traits that we derive from the microbiota. And what that gives us is a whole bunch of p-values that we can show like this as Manhattan plots. So now, as we go across the genome, so these are the chromosomes again with alternating colors, these are p-values derived from these regressions where we're regressing the IBD sharing against the quantitative data. In this case, you're looking at the abundance of proteobacteria, and you can see some very significant p-values here. So the higher the number here, the more significant the p-value. The Bonferroni correction line is shown. This is extremely conservative. But you can see that even some of these values are reaching the Bonferroni line. And if we look at what those are, for instance, this region on chromosome 11 contains some genes that have to do with epitopes on natural killer T cells. And so we are finding genes where the variation in the genome relates to the abundance of these proteobacteria, and these genes do make sense in that we would expect immune system genes to show up in these kinds of screens, because that's really the front line of interaction between the host and the microbiota. Here's another example for Bacteroides uniformis abundances. We see some high p-values for this region here. This is a gene that's regulated by NF-kappa B. It alters antigen presentation to T cells, and this particular gene has showed up in GWAS for Crohn's disease. So as we do more of this, and as we scale up to our full 600-person pilot study, what we expect to get is a list of genes and their interactions with microbiota that should hopefully confirm some of the mechanisms that we know underlie host microbial interaction in the humans, and then maybe open up some novel mechanisms to look into. Once we have our pilot study, we'll see about sampling the full 8,000, and then there's some other cool data sets that we expect to be able to integrate with, such as DNA methylation data, microbial data from other body sites that the Sanger Institute is generating. And then we'll be able to look at the effects of rare genes from these whole genome sequencing of 1,000 of the individuals. All right, now I'm going to shift to plants. This is a collaboration with Ed Buckler at Cornell, with JGI, especially Susanna Tringe and Phil Hugenholtz before he went off to Australia. And we work with Jeff Jangle's group at UNC. Aimé Spoor, a postdoc in my lab, and you might have met him yesterday at the poster session, has done a lot of this work with Jason Pfeiffer, who works with Ed Buckler. So just as the gut microbiota can profoundly influence health in humans, we know that the Rhizosphere microbiota can have a lot of different effects on plants. They can influence the immune stimulation, plant development, plant nutrient status, mineralization in the soil. They can influence plant hormone levels. They can influence soil structure, and so on. And so we wanted to look at how different plants influence the microbiota, and with the ultimate goal of mapping genes that could control microbiota composition in the rhizosphere. So just like we needed to see if the microbiota were heritable in the twins, here we're looking to see what heritable components there are in the root-associated microbiota by relating the variation in the root microbiota to genetic variation that exists between host lines. And we're using Ed Buckler's NAM system here that he's developed for corn that he'll talk about after me. So Ed started with a founding population of very, very different corn lines and derived these recombinant inbred lines that he uses to map plant genes that relate to phenotypes. We've been studying the parents because if we can't find differences between the microbiotas on the roots of the parents, there's not much point in going on to the big set of NAM plants. So what we've done is take these 27 of these founding lines and planted them in 5 different locations last year, this last summer, 2 in the Midwest and 3 in New York. And then we've been sampling 3 plants from each plot. We have a time series for the New York locations over 15 weeks. And then we've sampled the Midwestern plants at flowering time. We've also been collecting bulk soil samples. And so far we have 3 sets of experiments. We're doing this with this group of samples. One is we had to choose a primer that would minimize plant contamination, and I'll show you data we have from that. We're running right now all of the samples from the flowering time, and if the meeting was a month later, I could have showed you those, but they're coming off the machines at JGI now. And then we're working up the time series experiment data, so that's across 5 sites, and then 3 sites sampled weekly. We'll have about 5,000 samples to run on Illumina for those. The choice of primer was important. So we tested 4 different primers across the 16S region. There's different variable regions on a 16S gene, and these primers target different ones. So we looked at what they could pull up. And so combining all of the data, this is what we see. This is abundances of different bacterial phyla from our samples using the different primer pairs. And there was only really one that picked up very much plant material, and that was this one, 926 forward. And so this is the 20% of the reads that map to corn. But you could see that they all gave us pretty much the same profiles of bacteria, which was really good to see. There's a few differences. Some are more or less efficient at picking up some of these other phyla like Verrucomicrobia or Chloroflexi, and some can pick up Archaea because they're universal, like 515. And then, so what we've done is look to see if we can cluster the genotypes based on the microbiotas. So using Unifrac again. These are Unifrac dendrograms. Each one of these labels represents a sample. And if they cluster close together, it means they have more similar microbiotas. So here for the 27 forward primer, we see clustering by bulk soil. Here, a clustering of the 3 different genotypes we tested. And with the exception of this one outlier, which we actually think is a mislabeled sample probably, we see really nice clustering by genotype, indicating that that the different plant genotypes have different microbiotas. And we see the same pattern regardless of what primer pair we use. So here's the same pattern with 515 forward, and the same primer, the same pattern with this primer pair here. And this is these jackknife values tell you that this is robust to subsampling the data. Another way of doing this is to look at the abundances of particular taxa that we've detected. So here, data for 27 forward, you're looking at a heat map, if you will, of abundances of these different types of bacteria in the different samples. And now the samples are clustered based on whether or not they have similar profiles. And again, with the same outlier clustering with these here, you can see that we're separating out our bulk soil from our plant genotypes, and we're also separating out the plant genotypes. So regardless of how we analyze it, either using a tree-based approach or an OTU-based approach, we can tell our genotypes apart based on the composition of the root-associated microbiota. We can drill down into this and pick out specifically which taxa in the microbiota are discriminating the genotypes. So for instance, for this genotype shown in red, this genotype of corn, there's Chrysiomonas that's particularly associated with this genotype of corn. So as we go through the full set, we should be able to pick out specific taxa of bacteria that particular lines of corn are enriching from the soil. And the plan going forward would then be perhaps to sequence those representatives of those taxa. So we're finding that the choice of 16S primer can influence, to some degree, the taxa detected, but the discriminant OTUs that we pull out that discriminate between the plant genotypes are equally amplified by the different primers. So this is very robust to primer choice, which is really good. So the global pattern that we see of being able to differentiate the plant genotypes is also robust to primer choice. And we see specific types of bacteria that are being recruited by specific maize lines. So we've clearly got components of the root-associated microbiota that are heritable, at least from this initial study, and we should know a lot more about this by the end of April when we have our full 500 samples from across 5 fields. So our short-term goal here is to take a look at all the flowering time samples from the 5 fields, and then put in this time series component, because we have samples from 15 weeks. We'll be able to look at how time influences these patterns. We are planning on some whole genome sequencing of some of these important rhizosphere bacteria, and then also maybe some targeted metagenomics of some of these communities. In the long term, we're talking with Ed about sampling the particular NAM hybrids that would allow us to actually map the plant genes that drive this in the coming summers. And once we know that, then we should be able to, if we want, then think about incorporating this into breeding maize lines that are able to recruit specific types of microbes from the soil that might be desirable. So I'll end here. I'd like to thank especially Emmy Spoor in my lab, who did the vast majority of the corn work that you've seen, and Jason Pfeiffer and Ed Buckler, who've been instrumental for this, and then our collaborators at JGI and at UNC for their help with this project. On the human microbiome side, Andy Clark and Tim Spector and Julia Goodrich, the student who generated those data, have been really instrumental. We work closely with Rob Knight for bioinformatics support, and the work that I've shown was funded through NSF, the JGI, and the David and Lucille Packard Foundation. So thank you, and I'll take questions.
Thank you very much for an excellent talk. Questions?
Too early.
Actually, I have a question.
Okay.
First of all, I was at the International Microbiome meeting last week at Vancouver, and it was very interesting to see that one of the most oftenly asked questions was, what is the host response? And most of the speakers couldn't actually address that because they were not studied. So I'm very happy to see that. And the analogy also between the plant and the human system. So genetic background is one issue. The other issue is what is the differential expression of specific genes. So, for example, genes related with inflammation, etc. Are there any studies to see the response? The response under different microbial communities, the response of the host for specific genes like NF-κB, for example, or others?
Yes, most often in the context of these targeted gene or candidate gene systems. Yeah, so you start getting into immunology, but there's quite a bit of that within specific models. It would be much easier to do it on a bigger scale with the corn.
In relationship with specific microbiome communities. Okay.
So yeah, I'm starting to be sold on corn or plants as a very tractable system for this kind of thing. Yeah.
So I was wondering, in relative heritability, how heritable is the microbiome in humans compared to, say, Crohn's disease or some of these higher scale diseases? Is it more or less heritable?
Well, with Crohn's, there's particular mutations like in NOD2 that are really highly associated with Crohn's. So you've got a sizable proportion of people with Crohn's that have these mutations, for instance. So I'm not sure I could answer that off the top of my head in terms of heritability. But what I think needs to happen is that the microbiota need to be incorporated into these GWAS studies, so where you have variation in the host, variation in the microbiota, interactions, and then how those relate to phenotype. Because what I'm guessing is that there's probably particularly dangerous combinations, just like we've seen with H. pylori strains and gastric cancer risk, where there's particularly bad combinations of host genotype and H. pylori strain. That's probably true for some of these gut diseases, I would suspect. Yeah.
Would you show in a twin study correlations with certain genes that make sense? I would expect that should make a difference in the expression of genes, so more regulatory genes, transcription factors. It's probably early days for this
Don't know yet. We've just looked at a very small initial subset, but we should have a much better idea by this summer when we've ran through the full 600. So I can't tell you at this point. Yeah.
In your approach, you only target bacteria.
Mm.
It's well known that also fungi can protect plants.
Mm-hmm.
For a very important role. Wouldn't it be interesting to also see what fungi do?
It would be great. No, I agree. Yeah, and so the good thing is that we'll be banking samples and banking DNA extracts, so we can go back and do all these types of other measurements. Yeah. Okay. Thank you.